So thank you very much first to the organizers for the opportunity to participate. This has been such a wonderful workshop so far. I feel like I've learned a lot. I'm going to be touching on, I guess, some of the themes related to spatial and temporal heterogeneity, but more at a larger level, I'm going to be talking about some of the work that we've done on modeling the evolution of multi-drug resistance in HIV. And I guess we're actually going to step back further and think about drug resistance in HIV more broadly. And before I get started, I wanted to thank my co-authors on this work, especially Pluni Pennings, but also Dmitry Petrov and some of the things I'm going to talk about in the first half, and then Kristen Harper and Chancy Brum in some of the things I'll talk about in the second half. So I wanted to start by just giving you some of a sense of what is going on with the HIV epidemic. This is the number of deaths per 100,000 individuals in the United States, measured between the years of 1987 and 2016. And what we can see is that there's this rise in the death rate up until about 1995, and then there's a turnaround, and then we see this leveling off and sort of slow decrease of deaths related to HIV over this time period. And sort of knowing that this is a talk about drug resistance, actually, sorry, I want to make sure that I get a clock so that I'm staying on time. Thanks. Knowing that this is a talk about drug resistance, you may be looking at this and thinking, okay, well, 1995 has got to be the year in which we got our antiretrovirals. This actually isn't the case. The first antiretrovirals were FDA approved in 1987. And far from reversing this course of this increasing death rate, we see that they actually didn't do much at all in order to stem this increasing rate over time. And this is because, as I'll show you in a few slides, there was just a ton of drug resistance evolution happening in parallel across independent individuals within this time period who were treated with these monotherapies. What did happen in 1995 is we got sesquinavir, which is the first protease inhibitor on this list. This is a different class of drugs from the one marked in blue. And what it meant is that we had multiple different mechanisms of the viral life cycle that we can target. And so we could design combination therapies that meant that no single drug resistance mutation conferred resistance to all of the drugs in a particular cocktail. And it really was this as the sort of magic bullet that turned around the course of this epidemic and allowed us to treat HIV actively. So what I want to do today is just briefly take you through a few stories of what are the dynamics through which resistance emerges within individuals treated with ART, and try to highlight some of the things that we're still working on and some of the places where we still feel that, you know, mathematical modeling has a big role to play in terms of helping us better address this major problem in human health. So first, I'm going to talk about what was happening early in the pandemic when we were treating individuals with these monotherapies and they were basically instantly failing to drug resistance evolution. And then I'm going to sort of transition into addressing what has happened since then as we've started combining more efficacious combinations of drugs and hopefully give you a sense of what we still have left to learn and how we can explain how evolution continues to happen in the modern day. But just jumping into this first point, I want to give you a sense of how fast HIV can evolve in response to monotherapy. So what I'm going to show you is data from a trial of monotherapy treated individuals that was published in 1995. And on the x-axis, we have the weeks of treatment in a totally drug-naive individual. And on the y-axis, we have the percentage of the population that's drug-resistant as measured by large effect drug resistance mutations that can easily be captured from the blood. And so this individual was completely naive, not seen drugs prior to week 0. And what we see is that if you actually measure them over time, within a matter of weeks, the population goes from being entirely drug sensitive, as far as we can tell, to entirely drug resistant. So really rapid turnaround and fixation of drug resistance mutations. But it's not just that the evolution of HIV in response to a single monotherapy is fast. It's also extremely predictable. So I'm showing you 18 additional individuals who are all treated with the exact same therapy. And what we can see is sort of strikingly across these different individuals, resistance is fixing on basically the same timescale within all of these different individuals. So 18 of the 20 individuals I'm showing here had drug resistance at over 50% frequency by week 4 of treatment. So thinking about HIV generation times as being 1 to 2 days, this is, you know, conservatively full sweeps within, or, you know, major sweeps within 30 generations. So really fast, rapid evolution. And so looking at this plot, it sort of struck me that this is a parameter regime that isn't necessarily something that we think about often, that evolution can happen so quickly and predictably. So one of the first questions I wanted to address is how exactly is evolution happening so quickly and so predictably in these populations? And trying to derive things like estimating selection coefficients from time series data like this is actually one of the first things that got me excited about population genetics to begin with. But I actually want to back up and do something much stupider than that, which is just if we simulate a simple Wright-Fisher model with selection, how can we understand the data that I've just showed you? So I don't need to go through this in detail because I know that this is a common familiar territory for all you folks, but we consider a population of n viruses. Each generation, each virus has a probability mu of acquiring a drug resistance mutation. And so the total number of drug resistance mutations entering the population per generation is n times mu, or theta. And these drug resistance mutations confer a selective benefit 1 plus s relative to wild type. And what we can do is we can just look across a wide variety of the parameter regimes, looking across a variety of selection strengths and population mutation rates, and just do forward simulations and ask, under what sort of population genetic conditions in terms of theta and s do we see sweeps happening reliably in 30 generations, which is to say, 80% of the time or above. And if we run these forward simulations, what we see is that it's actually a relatively constrained part of the parameter space where you're able to get such rapid dynamics happening so predictably. So first, selection needs to be strong, otherwise sweeps can't reach high enough frequency rapidly enough. And the population mutation rate has to be sufficiently large that these beneficial mutations are entering the population so that they can be selected upon. And I'm going to draw your attention in particular to these selection strengths, which are, this is, a selection strength of 1, and this is not NS, this is S, meaning that really these are hugely, laughably large numbers in the context of something like population, human genetics. But we think that, this is really what we need to invoke in order to explain the sorts of dynamics that we're seeing in intra-host HIV populations. And it's worth stepping back and thinking about what rules get violated when we start thinking about patterns of genetic variation that look like this. And so I just want to sort of think about this picture that I showed you previously and think about what it would look like for a beneficial mutation to arise and sweep in a population like this. So under our conventional view, we imagine we have a change of environment, we have some waiting time for a beneficial mutation, and then when it occurs, it can sweep the fixation in what we all know is a hard selective sweep. In the context of intra-host HIV, we see that theta can get quite large. And so what that means is that our waiting time for beneficial mutation becomes small. And as this mutation is spreading in the population, the sweep time for that beneficial mutation may be larger than the waiting time for the next beneficial mutation. So we can see additional drug resistance mutations occurring even at the same locus and spreading in parallel. This is called a soft selective sweep. A lot of the work on understanding the theory of when and why and the genetic patterns of these soft selective sweeps was worked out by Pennings and Hermison in the mid-2000s. But the important thing to note here is that evolution is happening so quickly that it's actually happening multiple times before a single mutation is able to fix. And so if we think about what this means in the context of our really rapidly evolving intra-host populations, one of the things that we can see is that having a high probability of sweeping in 30 generations may give us some insight into whether those selective sweeps are hard or soft. And so what we can do is go back to these simulations and ask in each of these generations and each of these populations that did have a sweep, conditional on it having a sweep, what's the probability that that sweep was soft? And what we see when we check these simulations to see whether there are multiple beneficial mutations segregating above 15% frequency at the time of fixation is that there's actually the same sort of parameter regime where we're likely to see a sweep in 30 generations is similar to part of the parameter regime where the sweeps are likely to be soft. So what this is suggesting to us is that in the early days of HIV therapy, when evolution was happening so quickly that it was happening, with high probability within very short periods of time across multiple individuals, we can actually have some confidence that it was happening multiple times within the same individual. And this is something that we can actually go back and look for in genetic data. There are particular drug resistance mutations that make this particularly easy to find. So one example of this is this K103N mutation. It's a lysine to asparagine transition at the 103rd position of the Pol gene in HIV. And sort of importantly, lysine is encoded AA or AAG, and asparagine is encoded AAT or AAC. So what this means is that there are sort of two equivalent ways to encode an asparagine mutation. You can have an A to T or an A to C mutation at the 3rd position of this 103rd position. So one of my colleagues, Mooney Pennings, along with her colleagues, Sergei Krasimsky and John Wakely, did something that I thought was quite clever, which is that they went into a patient who was treated with a drug that was targeted by, the K103 and conferred resistance to. And what they did was they looked at this 103rd position across a bunch of different sequences sampled from this individual. And they can see at time 0, this first day before the individual saw any treatment, they're entirely AAA at this 103rd position. But by the time you sample them 28 days later, again, that sort of magical number of 30 generations, you see that you have a fixation of this asparagine allele or asparagine amino acid at the 103rd position. So at the amino acid level, you may look at this and say, this is a hard sweep of drug resistance. But if you look under the hood at the nucleotide level, you can actually see that it's comprised of both T's and C's, meaning there must be at least 2 independent mutations of, A to C and an E to T. So this is just one example, but this is a pattern that we can see across data more broadly, that within these monotherapy-treated individuals, sweeps are actually driven by multiple independent mutations conferring drug resistance. So what this is telling us is that early in the HIV pandemic, evolution was happening extremely rapidly and extremely predictably. And we think that at least some percentage of it was driven by these soft selective sweeps of multiple drug resistance mutations simultaneously. And in some ways, this is sort of not too surprising that this should happen. The population sizes are huge and the mutation rates are large. And I kind of like to think about the occurrence of soft sweeps as being something, intuitively related just to, say, a Poisson distributed random variable. If you can find a lambda that means that you're going to have a non-zero event with high probability, the probability of having 2 or more events within that pull from a random variable is going to scale equivalently. So, having a high probability of having at least one event means that you're going to have a higher probability of having 2 or more events, which is to say independent origins of drug resistance mutations. Okay, so this is not where we are today. Since then, we've made huge strides in terms of improving the rates of drug resistance. And I already sort of gave you a hint that the way that we've done this is by combining drugs into efficacious combination therapies that target multiple stages of the viral life cycle so that no single drug can confer resistance. I want to sort of put in a little side note that as these treatments have improved, we have seen a transition from soft selective sweeps to hard selective sweeps. And this is something that we can look for in genetic data by comparing the patterns of post-sweep diversity in a variety of individuals taken from, treated with things early in the pandemic and things with treated with things later in the pandemic. But I don't have time to tell you about this story in detail today. What I do want to move on to is thinking about how combination therapies are able to, how HIV is able to evolve resistance even under combination therapies. And we can see that despite the fact that drug-resistant rates have fallen pretty drastically over this time period, there still is a substantial amount or a non-zero amount of drug resistance evolution going on in the world today. And it's important to understand where this is coming from and ideally designing evolution-proof therapies that are not going to be subject to as much of resistance. So it maybe helps to step back and just walk through really quickly the logic of why we think these 3 drug therapies work. So if you have a virus and you treat it with one drug, the drug can acquire a drug resistance mutation and then it'll be able to replicate in the presence of the drug. Now, if you take a virus and you treat it with 3 drugs simultaneously, you have 3 different mechanisms that are blocking the virus from being able to complete a round of replication. Therefore, even if the virus gets a single drug resistance mutation and is able to replicate in the presence of the red drug, it's still blocked by 2 blue drugs. So we think it shouldn't be able to spread within the context of an intra-host population. What should need to happen is that a virus should need to get 3 drug resistance mutations simultaneously on the same genetic background to be able to complete a round of replication in the presence of 3 drugs. And we think the rate at which this happens should be significantly lower, mu cubed, say. Now, this is ignoring a lot of potentially complex dynamics that we'll maybe get to partially in a second, related to, say, stochastic tunneling. If this really is a really flat fitness landscape up until you get 3 drug resistance mutations, then maybe there's a possibility of these seemingly neutral mutations that don't help the virus complete a round of replication, sticking around long enough to get that second and third mutation. But in general, we think that this is a core idea behind why these therapies work so well. And from what we've seen so far, so it's worth thinking about what are the dynamics going on under the surface that can help us understand what's going on here. So to do this, it helps think about the concept of a dose response curve, which is some relationship between the concentration concentration of a drug and the ability of the virus to complete a round of replication. So what we think is that at high drug concentrations, or maybe let's say at low drug concentrations, the replication ability of the virus isn't going to be particularly interfered with by the drug that's present. So, this X gets small, and this goes towards R naught, which is some baseline R naught of the virus in the absence of the drug. But as X gets much larger, we see that this approaches zero, and the virus is able to complete a round of replication. And exactly the shape of this curve is going to determine whether the virus should be able to complete a round of replication or not. So what happens when you get a drug resistance mutation? At the sort of intermediate level, we have this critical R naught equals 1 line that we heard about in the last talk. So, if it's larger than 1, then the viral population will grow. And if it's smaller than 1, then the viral population will shrink. So hopefully we're treating with drug concentrations that are over here. So the viral population is shrinking. Now, when we get a drug resistance mutation, it changes the curve in a couple of ways. So the first thing it does is it moves the dose response curve over to the right. What this means is that there are higher drug concentrations that this virus is now able to replicate in the presence of. So we can see that reflected in this row term here. It can also change the shape of this dose response curve, which is not something that I'm going to talk about very much in the talk today. So it can also make it steeper or shallower. And then the other thing that it does is it can often come with a fitness cost of resistance, which we're seeing reflected in this 1 minus S term. So oftentimes at low drug concentrations, we can see that despite the fact that it's able to replicate at high drug concentrations, when the drug is absent, it's not able to replicate as efficiently as the wild type. So the dynamics between these two lines are going to determine whether we should see drug resistance evolve or not. So if we see that the drug concentration is over here, nothing should grow, both populations should shrink. If we see that the drug concentration is over here, because of the fitness cost of resistance, we actually expect that the wild type virus should dominate over the sensitive, over the resistant one. And it's really this intermediate mutant selection window that we need to be worried about, because this tells us that this is a place where the wild type is going to either not be able to grow or will not be able to grow as fast as the drug resistant type. And it's this particular window that we need to be worried about in terms of thinking about drug resistance evolution. And this mutant selection window is actually really useful for thinking about patterns from the Sherman data that I showed you previously. So if we think about, say, one of these trajectories in which we see drug resistance spreading within the population in a few weeks, and we look at what the viral load is doing during this period of time, we see that initially it starts high, it shrinks initially in response to the therapy, and then it sort of rebounds. And what this is telling us likely is that the wild type virus is sort of down here, unable to replicate replacement levels. And then when this drug resistance mutation emerges in the population, either from low frequency standing genetic variation or de novo mutation, and reaches high frequency in the population, we see the population recover. So it kind of goes from here to here, suggesting that we live here within the context of the dose response curve of the therapies that we saw in Sherman data. So the broader question then becomes, this thinking works pretty well within the context of a single drug. How does this work when we start thinking about combination therapies? Is this really, is this sort of logic that I laid out to you about 3 drug combinations, does this work here? And the challenge here is that you need to start thinking about how all of these different drugs are interacting with each other, and what are the different shapes and attributes of these different dose response curves, resistance mutations, and their corresponding mutant selection windows. So for example, even if we have a single drug, I'm not sure if you can see my pointer here on the right side, but even if we have a single curve that's in, say, the dose response curve with respect to drug C, if in the context of the presence of drug B, it's at very highly suppressed due to the presence of drug B and highly suppressed due to the presence of drug A, it's unclear that even if you had a C resistance mutation, whether that mutation should be able to spread, which is really the idea behind these combination therapies. So one of the things that I want to try to do is think about these combination dose response curves and see whether they could possibly select for resistance mutations and what sort of patterns we would expect to see. But before we do this, I want to do a brief sojourn into the data and ask, what does it actually look like when drug resistance evolves on triple drug therapy? Because this is something that I didn't have a lot of great intuition coming in to this project. And the more that I thought about it, the more I realized that it doesn't seem to match a lot of the patterns that we would expect under the conventional wisdom of why these 3 drug therapies work. So I'm going to present a series of surprises, or things that are sort of surprising to me under the conventional wisdom. And the first surprise is that actually drug resistance evolves at all. So we think that because the rate of the creation of this triple drug resistant mutant should be so low that we would expect it should be relatively rare. It should be extremely rare, actually, that these things should emerge. It should be a couple of cases per year if we're thinking about the number of instances of triple drug resistance mutations all occurring in a single round of replication. But what we see is that actually, despite the fact that triple drug combinations were introduced in 1995, there were still extremely high rates of drug resistance evolution for years after these 3 drug therapies were initially given. So one thing that we need to be able to explain is how is there so much ongoing resistance evolution despite the fact that we were giving individuals 3 drug therapies here? So one thing that maybe we have wrong is the rates at which these resistance mutations are happening. Maybe our mutation rate estimates are too low, or our population size estimates are too low, or there's something else going on, but the overall model is correct. So that's something that we should also be able to check, this idea that either we should see individuals with no resistance or individuals with triple drug resistance, but not really individuals with resistance to some subset of the drugs. And this is something that we can check in real clinical trial data. So I'm going back to another study from the mid-2000s, and where individuals were given a combination therapy of NFV, 3TC, and D4T. And so we can ask, among the individuals who were given this combination therapy, how many of them had no resistance to 1 drug, 2 drugs, or 3 drugs. And as we expected, we find that a lot of individuals are over here. They didn't get any resistance mutation as we would expect and hope. Now, if we look at the people who do have resistance mutation, the question is, do we find them all over here in the triple drug case? And the answer is no. We find many individuals that have a single drug resistance mutation or 2 drug resistance mutations. What this is suggesting to us is that resistance isn't actually spreading exclusively through triple drug resistance. It's more complicated in terms of how these mutations are acquired. Another thing that this plot is sort of suggesting to us is that it looks like there are these semi-predictable walks through this landscape where, this is not longitudinal data, this is a single snapshot. But it's sort of suggesting that first 3TC resistance is acquired, and then NFV resistance is required, and then triple drug resistance. And so, this is sort of suggesting that there are these semi-predictable pathways through this landscape that might help us figure out why these drug resistance mutations are emerging at all. This is a pattern we observe more broadly, that 3 drug combinations fail in a partially predictable order. So we went back into a large data set of individuals treated with triple drug therapies, and we looked only at individuals who were treated with particular combinations. So one that had a particular class of drugs, a protease inhibitor, and then 2 NRTIs, a 3TC or another NRTI. The precise details are not super important, but what we did is we subset these patients down to only individuals that had a single observable drug resistance mutation. And what we found when we did this is that overwhelmingly for these different types of therapies, 3TC was the first drug to fail among these therapies. So each of these blocks represents a different HIV population within a patient, and the color is based on which drug that single drug resistance mutation conferred resistance to. So we see that across these different therapies, most likely 3TC was the first drug to fail in these instances. We also did this for not just protease inhibitor-based therapies, but NNRTI-based therapies. And what we found is that overwhelmingly on NNRTI-based therapies, NNRTIs were the first to fail. So it was more likely that you were going to have both NNRTIs, even though 3TCs were possible drug resistance mutations, telling us more broadly that actually it's the combinations that are important in terms of determining this potential route towards failure. Okay, so you might be saying, all right, well, maybe these drug resistance mutations were pre-existing in the population. It wasn't just that they actually evolved on therapy. Maybe they were there before. They were either transmitted by an individual or they were present as part of standing genetic variation. If that's the case, then we would expect that most of the failures among these individuals should happen in the first year of therapy. So what we did is we looked at this large Canadian cohort and asked, and we tracked them over time. We didn't do this tracking. We looked at someone else's data set who had done this tracking, and we said, in what year of therapy did individuals get their first drug resistance mutation? And while we did find that in the first year of therapy, there was an excess of individuals acquiring resistance, there's actually still, a substantial amount of ongoing resistance evolution in, say, the 5th and 6th and 7th years of therapy. So this is telling us that even if someone has had no drug resistance for 5 years, there's still, say, a 2% chance, more than 2% chance of acquiring resistance mutation in their 6th year of therapy. So backing up, I want to kind of recap these 4 observations that I just told you about how combination therapy can fail. First, drug resistance still emerges under combination therapy. It does so via single mutations in a partially predictable order. and even years after therapy. So what we would like is a model that is potentially able to explain all of these different features that we're observing in real data to try to understand more broadly, the ways in which HIV is getting around these combination therapies. So there are a couple of different hypotheses that we were sort of considering when we were thinking about how these combination therapies could fail. So one of these ways is we're thinking, well, maybe the actions of these drugs aren't independent, even though we could imagine that maybe the drugs at the administration of the concentrations of the drugs in administration followed these dashed lines, and that, even though C was in its mutant selection window with respect to C resistance mutations, where we would think that C resistance mutation should have a good chance of spreading, if not for the suppression by B and A drugs. But one of the potential hypotheses here is that, A and B are not actually suppressing the virus nearly as much as we would think, because the drugs are targeting similar mechanisms, or they're competing for binding sites. It turns out that most of the drug interactions that we can measure pairwise between combinations of drugs are not antagonistic. I don't want to go through the slide in too much detail, but essentially we could imagine that there are instances in which two drugs combine in a way that is essentially multiplicatively, so that the action of each of the drugs is totally independent with respect to each other, in which case we would expect the suppression value of the two drugs combined to be multiplicative with respect to the two drugs measured individually. We could also imagine there's some instances in which they're more behaving as additive combinations. So they do suppress, but it's not nearly as strong as when we have two drugs considered together. Or we could consider that there are also instances in which the combination of two drugs are actually more suppressive than the two drugs considered multiplicatively. But if this group led by Bob Silicano went back and did all of these measurements, and they found that actually for the most of these drug combinations that we would consider, each of these are a different HIV drug, the synergy or the combination of the two drugs is not antagonistic. So we don't see a lot of these red lines indicating that the two drugs are performing worse together than we would expect. And in fact, the three therapies that I showed you in combination earlier in the KEMF trial, D4T, NFV, and 3TC, are all acting sort of either nearly bliss independently or almost bliss independently. So what this is suggesting to us is that the reason that we can see the emergence of drug resistance isn't because the drugs are just not very effective when they work together. It seems like they're actually quite effective when they work together. So what I do want to consider is potential hypotheses, sort of in relation to a lot of the themes that we've already talked about in this week so far, are how these selective pressures are varying in space and in time. So we can think about these two hypotheses sort of separately and then try to understand whether we can they can potentially explain the patterns that we see. So one of them would be that there is temporal heterogeneity of the drug due to a decay in the drug levels after dosing. So we could imagine what the drug concentration over time looks like in an individual who's taking these pills. So of course, individuals don't have totally constant drug concentrations over time. They fluctuate throughout the day as people take therapy, and that drug is metabolized over time. So we imagine after a particular dose, we start at some high drug concentration that decays according to some half-life. And these are well measured within the context of HIV antiretrovirals. And we can think about what the replication ability of the virus, a totally sensitive drug virus or a totally drug-sensitive virus versus one that's resistant to this A drug, this B drug, or this C drug are over time. And we can see that as the drug is decaying, the replication ability sort of goes up a bit. But then, you take another dose of therapy and the drug concentrations shoot up again, and the replication ability of virus sort of leveled out at zero again. So over time, these drugs are dosed in such a way that the replication ability of the virus should never reach this line. It should never be possible that the sensitive drug or even any of the single drug resistance mutations should be able to spread, ideally. But if an individual misses a dose, then the drugs can continue to decay over time. And with a sufficient number of doses, we may enter in a time when drug concentrations are washed out enough for specific subsets of the drugs that single drugs may have entered their mutant selection window without the additional suppression of these other drugs based on these single drugs, the additional suppressive ability of these other drugs. However, if the individual keeps missing therapies, we might imagine that there's another transition away from the state where a single drug resistance mutation can be selected and into a state where the sensitive virus is actually the one that dominates. And of course, if you take drugs again, then the ability of each of these viruses to replicate shoots down enormously again. All this to say that it's possible to imagine scenarios in which these single drug resistance mutations are able to spread, but it's complicated by the fact that it depends very narrowly on having drugs in the correct drug concentration windows. And it's not clear whether for HIV therapies, whether they're maintained in these windows for large periods of time. Another hypothesis is that there's heterogeneity of the drug concentration with respect to space. So we may imagine it's really easy for the drug to get into the blood, but it may be harder for the drug to get into the central nervous system and across the blood-brain barrier. And this is something that's been measured empirically within the context of ART concentrations within individuals. So the idea behind this is very similar, just that there are some areas that are more difficult for the drug to access and some areas that are easier for the drug to access. And while I've drawn these as sort of a cartoon of nesting things where, drug B has strictly dominant penetrance as concerned to drug C and drug A, in reality, there could be more complicated patterns in which, drug A is good at getting into the lymphatic system and drug C is good at getting into the central nervous system. But the idea here is the same in that there may be areas of the body where most of the body, there's no replication that's able to happen because of the drug concentrations, but in certain areas, we may imagine that a single drug resistance or the drug-sensitive virus is able to spread. So the question is, can these two different models of spatial and or temporal heterogeneity explain the patterns that we've seen in the data? So is there ongoing resistance under these models? Is there one mutation at a time? Is there a predictable order? And does it happen even years after therapy? And a lot of these different things seemingly could fall out of this model. In particular, this predictable order may be happening based on which drug is the one that sticks around in the blood for the longest, has the slowest half-life, so is likely to be present even after the other drugs have decayed, and which drugs are the most penetrant, where are they able to get into the tissues the most effectively. And it's those drugs that we should think should be the first ones to fail within the context of therapy. So what we're going to do is look at this particular combination that I showed you earlier and ask whether these patterns can plausibly be explained by combination therapies, heterogeneity of combination therapy in space or time. So our model of HIV intrahost drug resistance evolution is as simple as we can make it, which is just that we have genotypes evolving as bit strings, where we think that there are certain mutation rates at each of these three potential drug resistance sites. And so there's a certain mutation rate of acquiring drug resistance for each of the three drugs. And based on the mutational target sizes and the genetic composition of these different sites, we can actually measure what these mutation rates are directly. And then what we can do is we can compute the R0 for each of the viral genotypes for a given set of drug concentrations, 3T, D4T, and NFV. So namely, an entirely drug-sensitive virus to an entirely resistant one. And the way that we get these Rs are by looking at these empirical measured drug concentration dose response curves for these three drugs in question, 3TC, D4T, and NFV. And so what we can do is for any given combination of each of these three drugs, we can ask what is the ability of each of these different genotypes to complete a round of replication. So how do we figure out what the concentrations of the drugs that we care about are? Well, we model a temporal heterogeneity model and a spatial heterogeneity model. So under our temporal heterogeneity model, we imagine that the drugs start at some level, which is the dose, the Cmax that we think is administered at initial administration of the drug. And it's decaying over time according to the half-lives of these drugs. And if an individual misses a dose, which each individual does with probability P, there's an underlying distribution of probabilities of missing a dose. We see that the drugs can continue to decay over time. The second thing that we do is, another important thing to note about this temporal model is that there is able to actually be total viral exhaustion under this model if someone is very adherent, but we think that in HIV, this doesn't happen because there's a latent reservoir with cells that continue to wake up even after someone has been on therapy for long periods of time. And so the viral population is continually refreshed by this latent reservoir. And we think that this latent reservoir has drug concentration or has the frequencies of drug resistance mutations that are seeded according to mutant selection balance before therapy. So if you have a really costly resistance mutation, it should be rare within the context of therapy or within the context of the reservoir. And so our model of spatial heterogeneity assumes it's, again, much similar than a lot of the models that people are presenting here in terms of spatial structure, but we assume that there's a large component of the body that's proportional to the size of the blood plasma, where they're getting full doses of the drug measured over measured over a complete dosing cycle. We take the average. But then we also model a smaller central nervous system compartment that is concentrated, or that has a concentration that's proportional to measurements of the central nervous system blood depletion. Or sorry, the concentration of these antiretrovirals that are measured in the central nervous system versus the blood. And so what we see is that the drug levels are substantially lower within the central nervous system, and we model this based on the size of the cerebrospinal fluid, the number of liters. And then we finally have a small sanctuary compartment which corresponds to the latent reservoir as in this model. And what we can do is simulate under these two models and ask whether there's temporal heterogeneity of under models of temporal heterogeneity versus spatial heterogeneity. We see these same types of patterns. And so we run stochastic simulations for up to 5 years of therapy. And anytime we have individuals who are basically simulating experimental conditions from the CHEMP trial where they come in every 3 months. And if their viral load is above a certain threshold, we assay them for large drug resistance mutations in order to get a sense of whether any resistance has been emerging. And so I'm going to show you the results from the temporal heterogeneity trial. What we namely see is that actually we are able to evolve drug resistance under temporal heterogeneity, but largely only to the drug 3TC. What we see is that almost all of this resistance happens in the first year of therapy. And the reason for this is because if you have an underlying probability of taking your adherence probability, if it's sufficiently low that you're selecting for resistance, which is to say, if it's too low, it doesn't select for resistance. If it's too high, you never get a chance for resistance. But if it's in this sort of sweet spot for resistance evolution, you get it relatively quickly. And then afterwards, there are relatively rare instances of resistance happening after that. On the other hand, if we look at our model of spatial heterogeneity, because there's this ongoing chance for individuals to be entering this compartment where there's an opportunity for the selection of these intermediate resistant phenotypes, what we see is that there's actually substantially more ongoing resistance evolution even years after therapy. We can also check some of the other patterns, namely, we see that in most instances, individuals have 1 or 0 resistance mutations at virologic failure, and we see a lot of individuals that are resistant to 3TC versus the other 2 drugs, which again is apparent from these plots. So in aggregate, what this is telling us is that some of the, while many of the patterns are able to be matched by a model of temporal heterogeneity, some of this key crucial one about ongoing resistance evolution happening even years after therapy seems to be better matched by spatial heterogeneity than temporal heterogeneity. Although there's some chance that individuals are drastically changing their adherence level even years after therapy, which could be some reason why we see this, why it's possible that temporal heterogeneity can explain this as well. So I want to just mention that a lot of the parts of our model are extremely simple, especially the spatial and temporal heterogeneity, taking drugs, with a certain probability, or, just having these 3 compartments. And we're looking to try to improve on some of these things, namely by taking measurements of drugs at really modular, or really specific concentrations within the context of single organs. So I have a graduate student who's been working on simulating T-cell movement through organs, in which we have really high resolution measurements of the concentration of given drugs, this meroviric, over time. And what he's observed is that you can have really different experiences of drugs based on where individual T cells are moving within the context of these tissues. So, if you're up here, you may have you may be seeing very little drug, whereas if you're over here, you may be seeing a whole lot more drug. And so this is something that we're trying to incorporate into models, figure out how to go with, continuous space instead of this sort of caricature of these 3 compartments. We're also trying to incorporate more realistic models of adherence by using things called data from Y-spill devices, which make measurements exactly when they're opened, so you can get a better sense of when real individuals are actually taking drugs, which are likely to follow much more complex patterns than what we've modeled here. So I want to sort of wrap up because I'm out of time, which is just that under combination therapies, we think that potentially spatial or temporal heterogeneity could be a way in which resistance is able to emerge, but this is very much a work in progress where we're still trying to figure this out. We've explored some of these simple therapies for which we know a lot about the dose response curves and the concentration windows, but we have a lot still to learn here. And thinking about this more broadly, we have all of these different combinations of different drug types, and figuring out good ways to combine these into therapies that are really resistant to resistance evolution is a really promising way to think about how mathematical modeling could be harnessed in order to try to help us get a sense of, actually having real impact on people who are dealing with being impacted with HIV. Okay, so with that, I want to again thank my collaborator. Clooney is my main collaborator on a lot of this work, but also Dmitry Petrov, Kristen Harper, and James Brum. My funding sources, I want to thank the organizers again for the opportunity to speak, you for your attention, and I'll just do a final plug that I'm recruiting students and postdocs, so any of this is exciting to you, I encourage you to get in touch. Thanks again.
Thank you. So other questions with real people? I don't see questions yet on Zoom.
Yes, we have questions.
Okay.
Thank you very much. It was really a fascinating talk. I really enjoyed it. Thank you very much. I have two questions for the spatial structure. I think you have like those different compartments with different conditions of drug densities. We could imagine two other things for the impact of spatial structure. One would be a delay, like you would have propagation through tissue, and this could maybe add a new feature. And the other one is instead of having only a mutation that gives you complete resistance, have something progressive. I don't know if this could make sense biologically.
Yeah, those are both great suggestions. So towards the first one, the idea that we think that the penetrance is progressive and that there are there's a time delay in terms of where and when these drugs are penetrating. I think that's a really important thing to keep in mind. And the way that we've modeled it so far has been honestly really kind of everything is so simple because we're struggling with the complexity of the system. So we think that actually in terms of delay, there very well could be, first the drug hits the main plasma compartment and then only later is able to migrate into the central nervous system. But in another way, we could also imagine that the decay dynamics in the central nervous system are really not the same as what they are in the plasma. And that's something that we also haven't considered. So in short, I love that suggestion. I have not done anything with it yet. Towards your second question about the patterns of drug resistance in terms of not single full effect drug resistance mutations. So I think that is captured to some extent in these dose response curves where in some instances we see, like I think one of the major reasons we see 3TC evolving as the first mutation is because it has really strong large effect drug resistance mutation that has a tremendous cost, a much larger cost than some of these other ones. However, the rescue effect is much greater than sort of like D4T, which is really kind of a rubbish drug resistance mutation, all things considered. So right now we've only created these composite drug resistance mutations that grab the most sort of the strongest feature from each of these different categories. So in terms of the effect on the dose response curves, the largest selection cost, the greatest increase in IC50. But you're right that trying to model the entire spectrum of all drug resistance mutations that are available, and a lot of these have been measured empirically, is a great actual next step as well.
Thank you very much. Thank you.
Okay, thank you again for a great talk. I had a question about other mechanisms to generate this resistance, and I don't think you talked about things like recombination. If there is some, for instance, some multiple infection of same cell by different mutants, does that actually happen in real life, and could that be a way to explain fast adaptation?
Yeah. So the recombination rate in HIV is very high, and yes, it's something I'm also very interested in. Recombination, we do know, is a major factor. So the mutation rate in HIV for transition mutations is somewhere on the order of 10 to the negative fifth per base pair per generation, and the recombination rate is estimated only to be same order of magnitude, also 10 to the negative fifth. Suggesting that recombination is a major factor here. You bring up another important factor, which is co-infection. You need to get two viruses into the same cell in the same location in order to have that recombination event occur. So one thing that I'm really interested in is sort of what is the overlap zone of these different drug concentrations if we really do think a spatial model could be relevant. Because in that case, so one of the key questions we might imagine is if you get one drug resistance mutation in one area and one drug resistance mutation to another area, how do you get those two resistance mutations into the same cell for a recombination event? happen? What does the basically hybrid zone need to look like in terms of the overlap of these different selection pressures? This is something that a student in my lab is working on, just in terms of thinking about things like viral density in terms of the probability of recombination. And her preliminary results are that it seems like there's a lot of recombination regardless of what the viral load is, but this is something that we're still working on tracking down. Recombination is totally important, and we haven't considered it at all in this model.
Thank you.
Are there still questions in real life? Or
Thank you very much for your talk. If I understand well, you have stated that single mutation could trigger multiple resistance in once. And wouldn't there be a link with the fact that the specificity of HIV virus, which is very compact, to the extent that one single nucleotide may be used in 3 different codons? So, I mean, one basis changes and it may make 3 genes which are changed. And wouldn't there be something important in the mutability and the possibilities in mutations?
Yeah, so I think I maybe wasn't totally clear about this. So we think that no single drug resistance mutation confers resistance across classes of drugs, although that can happen within a class. So maybe if I go to the slide. So if we look at these different classes, each of these targets a different mechanism, and largely we think that mutations that confer resistance to the yellow drugs don't confer resistance to the drugs, and so generally what we do is we combine multiple different classes so that no single resistance mutations hit all of the drugs, although there are resistance mutations that confer resistance to multiple things within the red class of drugs. I wasn't totally clear that we think that the resistance mutations are non-overlapping between the classes.